Hello and welcome to the Biological Anthropology lecture series. I'm Dr. Richard Jonathan O. Tadurin. Here we explore what it means to be human, biologically, evolutionarily, and behaviorally. Let's begin our journey into the science of humanity. Welcome to Module 2, the biological basis of evolution. Here we explore cells, DNA, inheritance, and the evolutionary processes that shape all life, including humans. Before we dive into the big ideas of evolution, let's slow down and revisit the tiny structures that make it all possible. Think about this. Every feature you can observe in a living organism, eye color, height, disease resistance, even patterns of growth, begins at the cellular level. Cells carry DNA and DNA carries information. Understanding that flow of information is key to understanding how evolution works, because evolution operates on variation, and variation begins in our genes. Let's look at cells and chromosomes as the basic architecture of inheritance. You might wonder why this matters when we talk about human origins. Well, every evolutionary change you've ever heard about, whether it's the emergence of bipedalism or differences in skin pigmentation, can be traced back to changes in DNA, how it replicates, and how it's passed on. DNA isn't a static instruction manual. It copies itself, it makes mistakes, it recombines, and it interacts with the environment. These small molecular events ripple outward across generations, shaping populations and, ultimately, species. When we follow these processes from replication to protein synthesis to genetic variation, we begin to see evolution not as an abstract theory, but as a series of observable, measurable biological mechanisms. As we move through this module, keep asking yourself, how do small changes at the molecular level scale up into the vast diversity of life we see today? That's the journey we're starting here. In this section, we begin with the building blocks of life: cells, DNA, chromosomes, and the mechanisms of inheritance. Let's begin by grounding ourselves in the simplest question: what is life made of? First, let's revisit the idea that every organism, whether it's a bacterium, a banana plant, or a human, starts with cells. Cells are the smallest units that can carry out the processes we recognize as life. They take in nutrients, convert energy, repair themselves, and pass on information. Think about this. Everything that becomes a human being begins as a single cell, and from that starting point, division and specialization create the trillions of cells that make up the body you're using right now to watch this lecture. Now, let's look at the two broad categories of cells you'll encounter in biological anthropology. Prokaryotic cells, such as bacteria, are structurally simple and lack a nucleus. Eukaryotic cells, including all plant and animal cells, contain a nucleus that houses DNA and a set of organelles that perform specialized tasks. You might wonder why this distinction matters for our field. The answer lies in complexity. Human evolution is tied to the increasing sophistication of eukaryotic cells, especially their ability to manage and protect large amounts of genetic material. Within the human body, we also distinguish between somatic cells and gametes. Somatic cells make up your tissues and organs and contain the full diploid set of chromosomes. Gametes, sperm and egg cells, carry only half that number. Without the reduction of chromosomes in gametes, inheritance would quickly become chaotic. The careful halving and recombining of genetic material during reproduction keeps each generation stable while still allowing variation to emerge. Let's think about chromosomes as the organized packages that make this possible. In somatic cells, chromosomes come in matched pairs, one from each parent. They're not random strings of DNA. They're structured, tightly wound units that ensure accurate replication and transmission of genetic information. When cells divide through mitosis, they create identical copies of themselves, supporting growth and tissue repair. When gametes form through meiosis, the process introduces variation through mechanisms like crossing over and independent assortment. As we move forward, keep this idea in mind. Cells are not just tiny building blocks. They are active, dynamic systems carrying the instructions that connect one generation to the next. Understanding how they function gives us the starting point for everything else in biological anthropology. Let's take a closer look at the molecule at the heart of inheritance, DNA. First, let's revisit why this matters for us in biological anthropology. Every trait we study, whether it's a skeletal feature, a pattern of growth, or a population-level variation, traces back to information stored in DNA. So understanding what DNA is and how it works gives us the lens to understand how humans change, adapt, and evolve. DNA, or deoxyribonucleic acid, is a long, twisting molecule shaped like the famous double helix. Think of it as a spiraling ladder. The sides of that ladder are made of sugar and phosphate, and the rungs are pairs of nitrogenous bases. Now this part is important. Those bases, adenine with thymine and cytosine with guanine, pair in a very specific way. That strict pairing is what lets DNA store information reliably and copy it accurately. It's like having a built-in quality control system at the molecular level. Let's look at how that structure translates into function. DNA sits inside the nucleus of eukaryotic cells, tightly packaged into chromosomes. That packaging isn't just for organization, it protects the molecule and makes it possible for enormous lengths of DNA to fit into each cell. You might wonder how enormous. If you stretched out the DNA in just one human cell, it would be about 2 meters long. Yet it's coiled so effectively that it fits inside a nucleus you can't even see with the naked eye. Another key idea is that DNA is not a single uninterrupted strand of important information. Only a small fraction codes for proteins. Much of the rest is regulatory or involved in controlling when and how genes are expressed. This helps explain why two organisms with similar numbers of genes can still end up very different. It's not just the ingredients, it's the timing, the instructions, and the regulation. Now let's think about replication. Every time a cell divides, it needs to pass on an exact copy of its DNA. The molecule unwinds, the base pairs separate, and each strand serves as a template for building a new complementary strand. Because of the base pairing rules, the cell can recreate its DNA with remarkable accuracy, but, and this is where evolution enters, the process is not perfect. Mistakes or mutations sometimes slip through. Most do nothing, some are harmful, and a few introduce changes that can be favored by natural selection. This is how molecular events scale up into evolutionary consequences. It's also worth noting that not all DNA behaves the same way. Mitochondrial DNA, found outside the nucleus, is inherited only from the mother. It mutates at a steady rate and plays an important role in tracing population histories. One reason it appears so often in anthropological and forensic research. Keep thinking of DNA not as an abstract molecule, but as a dynamic information system, one that copies, mutates, regulates, and ultimately shapes the biological diversity we see in humans today. Let's turn now to one of the most elegant processes in biology, how DNA replicates and how chromosomes make inheritance possible. First, let's revisit the idea that every cell in your body, except your gametes, contains the same complete set of genetic instructions. For that to happen, cells must copy their DNA with extraordinary accuracy each time they divide. Without reliable replication, growth, repair, and reproduction would be impossible. Let's look at how that copying actually works. Imagine the DNA double helix as a twisted ladder. When replication begins, enzymes unwind the helix and separate the two strands, almost like unzipping a zipper. Each strand then serves as a template. Because adenine always pairs with thymine and cytosine always pairs with guanine, the cell can rebuild the missing half with remarkable precision. You might wonder why this matters beyond the molecular level. The answer is that faithful replication preserves the integrity of the genome, ensuring that the instructions for building and maintaining the organism stay consistent across billions of cell divisions. But DNA replication is not perfect, and this is where it gets interesting for anthropologists. A rare copying error, a mutation, can introduce a new variant into a population. Most of these changes go unnoticed. Some are harmful, but occasionally, a mutation appears that offers an advantage under certain environmental conditions. Those small chance events ripple across generations and become part of the evolutionary story we study. Now, let's shift to chromosomes because replication alone doesn't explain how genetic material is organized and transmitted. Chromosomes are tightly packed bundles of DNA and proteins. Think about this. If DNA were left loose inside the cell, it would tangle and break easily. Instead, it's coiled into structures that protect it and help the cell manage enormous amounts of genetic information. Humans typically have 46 chromosomes in somatic cells, arranged in 23 pairs. One chromosome in each pair comes from the mother and the other from the father. This pairing is essential for inheritance because it ensures that genes are passed on in a balanced and predictable way. Not all chromosomes function in the same way. Most of them are autosomes, which carry the majority of the genes responsible for everyday biological functions. But the last pair, the sex chromosomes, plays a different role. XX results in a typical female pattern of development and XY in a typical male pattern. What makes the Y chromosome fascinating is that it carries relatively few genes but has a crucial determinant that triggers male development. Let's look at how chromosomes behave when cells divide. In mitosis, which happens in somatic cells, the goal is simple: create two identical daughter cells. The chromosomes replicate, line up, and are pulled apart so each new cell gets a complete set. This process is essential for growth and tissue repair. Meiosis, however, is a different story. This specialized division creates gametes, eggs, and sperm, and involves 2 rounds of division. Here, chromosomes pair up and exchange segments in a process called crossing over. This shuffling of genetic material creates new combinations of alleles, which is one of the main generators of genetic variation. Then, during separation, each gamete receives only 1 chromosome from each pair. You might wonder why this reduction is necessary. Without it, chromosome numbers would double every generation, quickly becoming biologically unsustainable. As we move forward, keep in mind that replication and chromosome behavior are not just cellular mechanics. They are the foundation for inheritance, variation, and long-term evolutionary change. Everything we trace in human biology, from family resemblance to global patterns of diversity, begins with these molecular events playing out inside every living cell. Let's revisit the journey we've taken in this section. We started with the cell, the fundamental unit of life, and followed the trail inward to chromosomes and the DNA they carry. Along the way, we looked at how these structures organize, protect, and transmit information with astonishing precision. You might wonder why we spent so much time on processes that happen far beyond what we can see. The reason is simple. Every biological pattern we study, from individual development to human evolution, rests on this microscopic foundation. Think about how these pieces connect. Cells provide the stage, DNA offers the script, and chromosomes manage the distribution of that script across generations. Replication ensures continuity, while meiosis and mutation introduce variation. Now this part is important. Variation isn't just a detail. It's the raw material of evolution. Without those small differences created during replication or gamete formation, populations would remain genetically static, leaving no room for natural selection to shape them over time. Let's look at this as a story rather than a list of processes. Each generation copies a blueprint, but with slight changes, some intentional through recombination, some accidental through mutation. Those changes accumulate, interact with environments, and influence which traits persist. What began at the level of molecules ultimately shapes human diversity, adaptation, and evolutionary history. As we close this section, keep the big picture in mind. Understanding cells and DNA isn't just background knowledge. It's the key to understanding how life builds itself, maintains itself, and transforms across deep time. Thank you for joining this lecture. I'm Dr. Richard Jonathan O. Tadurin. I hope this session deepened your understanding of biological anthropology. Keep exploring what it means to be human. See you soon.